Okay, so as promised, I'm gonna go over to practice midterm. I went to Allison's office hours today. And again, guys, sorry these videos are coming up so late. I really did try this week to juggle all the classes, juggle studying, and juggle making videos, but recording them takes a while. No, actually, no, recording takes about an hour each time, but it's the Adobe Premiere editing, the rendering, the uploading, that takes a while. I really hate UCSB's upload speeds here. I mean, it makes sense. So, well, let's not waste any more time and let's just jump right into it. So, question number 1 is shown below our diagrams illustrating the shapes and relative sizes of blah blah animal viruses from 3 families. The genome of one of them is a DNA virus, the other 2 are RNA viruses. What is virus family to which each likely belongs? Alright, so this one is pretty straightforward. You have your A right here. You can probably guess that, it's, dang it, it's enveloped. And you count the segments right here, there's 8 of them. So what virus do we know is enveloped and has 8 segments? Influenza. So put influenza right there, or you put orthomyxoviridae. B, we could take a few guesses. We could say picornaviridae. We can say Rioviridae, Polyomaviridae, Papillomaviridae, but we don't just quite know what it is, because it's just so many icosahedral naked viruses that we've learned already. So maybe we should do B later. So we do C next. There's a bullet. Okay, so what is that? Obviously, Rhabdoviridae. Now we know that flu and Rhabdoviridae are negative single-strand RNA, so that rules out all these. Okay, so going back to B, what is B? Is it picornaviridae? No, because that's an R. We've already used up our 2 options of RNA genome viruses. So we can't say picornaviridae. We can't say Rioviridae. There's only 2 other options left. Polyoma and papilloma. So either of those two choices were for B. So A should be flu, B is polyoma or papilloma, and then C is Rhabdoviridae. This question right here is asking, now out of these 3 that we've labeled, where on this growth curve are they? So this is actually one of the handouts that Dr. Sears provided, and if you recall from that handout, it's one of the handouts from week 2. Recalling from that handout, we know that picornaviridae and Rhabdoviridae are 2 of the fastest growing viruses. So this top line right here, these huge, this is Rhabdoviridae. Recalling from that handout again, Paramyxo and orthomyxoviridae have the same growth curve, which is this middle one. So this is influenza. Now this is where it gets kind of confusing, because on the handout, this third one right here is shown. Oops, he only shows Rioviridae, but through a process of, we don't have Rioviridae from part A. So you can't put Rioviridae here, but through a process of elimination, you can put polyoma or papillomaviridae. And it kind of makes sense because it's a double-stranded, whether you choose polyoma or papillomaviridae, they are double-stranded viruses, so I assume that they take a while, they take a bit longer to grow than others. Okay, so the question number 2, the study of SC40 has taught us a lot about how genes work in animal cells. Now answer questions. The difference between permissive and non-permissive cell in the context of outcome infection is with SC40. Give an example of each, and tell what cell line the examples come from. Now, I personally would also go a bit into the results of using permissive cell and non-permissive cell. So the answer that I put down for that was an example of a permissive cell is CV1. which is a monkey cell line. And if you infect a monkey cell line with SV40, then you get infectious progeny. It's a productive infection. An example of a non-permissive cell line is 3T3, which is non-permissive. It's a mouse cell line, and when you infect it with SV40, you don't get productive infection. You don't get viral progeny. You get transformation of the mouse cell into a tumor cell. Question B, the difference between early and late transcript gene expression in context of temporal reference, blah, blah, was the temporal reference point, was the functional nature of the early and late protein products. Okay, so this question is testing your knowledge testing your understanding of what a temporal control point of gene expression is between a spatial control point. So the thing is, viruses, they have to regulate what genes to express and when, and they can either do that and there are two ways that they control it, and they use both. When we say temporal reference point, usually that's like in the context of the timing at what stage of the infection the virus is in. So like if it's like at the uncoding phase and transcription translation phase, then the early genes would be expressed. If it's now past that stage and it has gone through RNA or DNA replication, then it will do late gene expression. So for this question specifically, SV40, its temporal reference point is whether or not the virus has replicated its DNA or not. And then what's the functional nature of the early innate protein products? Well, the early protein products are your T antigens, and the T antigens drive the host cell into the cell growth cycle, so that the cell can make DNA and protein products, and not just any DNA and protein products. It will actually make the virus DNA and protein products, because the virus has tricked the host cell into growing DNA and growing protein, and tricking the cell into growing the virus versions of that. The late protein products are just capsid proteins. Now, just as a side note, if you want an example of a spatial reference point in the control of early innate gene expression, excuse me, think of human papillomavirus. So with that, papillomavirus controls whether it expresses its E or L genes, its early or late genes, by what cells it is actually in. So if it's in your, like for a woman, if the HPV is in in an undifferentiated cervical cell, then it will express its early genes. But the moment it gets to like the stage, once the cell gets to the stage at where the cell is differentiated into like a true cervix cell, it's like at the tip of your cervix and so forth, now the SV40 will express late genes. So good side note there. Question C, difference between, where's my mouse? There we go. Difference between 21 base pair repeats and 72 base pair repeats in context, the mechanism, blah, blah. You can read the question yourself. Okay, now, remember this isn't, so SV40 has its, its RNA it has its gene, right? It has its gene, and that gene has a TATA box, but also has a 21 base pair repeat, right? And there are 3 boxes of that 21 base pair repeat. And then there are also 2 enhancers, and those enhancers are 72 base pair repeats. Now, 21 base pair repeat, we said that there are three sets of those 21 base pair repeats. So I'm answering your question right now. Each of those 21 base pair repeats, each one of those boxes have 2 boxes of GC rich sequences. Now, with the 21 base pair repeats, what happens is, should I do this from memory? Gosh, I had this too. SP1, so specialized protein 1, binds to the 21 base pair repeats. And when SP1 binds there, the function of 21 base pair repeat is that it tells the cell, it tells the cell whether or not to express the early or late viral proteins. So if SP1 binds to the first 3 boxes of GC rich sequences, then you get early gene expression. If it binds to the last 3 boxes of the last 3 GC rich sequences of the 21 base pair repeats, then you get late gene expression. Now for the 72 base pair repeats, that functions as an enhancer, and an enhancer basically increases transcription in a way that transcriptions increase through the 72 base pair repeat is that a protein called AP1 binds to that area. Now, when AP1 binds to that area, it stabilizes the RNAs that are made, the RNA transcripts that are being made. And so when you stabilize the RNA transcripts, you get higher transcription level. Okay, so this, I'm not going to go too much into detail for it. I think this one's pretty self-explanatory. So I'll just read off the answers. Some of these questions we haven't gone through in class yet. This is a practice midterm. Obviously, some questions, if it's not something we need to know, I'll just say ignore, and that means don't worry about it. Okay, but so let's begin. So for A, the answer is 6. B, ignore it. C is 6. D is 6. E is 7. F, ignore it. G is 8. H is 5. I is ignore I. J is 2. K is 2. L is 3. M is 2. N is 3. O is 3. Okay. Now on to question 4. So basically, hypothetical situation. You're doing what I've always wanted to do but couldn't. You're undergraduate researcher, and you find a new strain. Hooray! So you know that this strain UCSB 2001, it can be grown in a raccoon cell, and that raccoon cell is permissive, and it's a papovavirus is another name for polyoma viridae. This used to be the original name for polyoma viridae, but then it changed to polyoma viridae. So you know that this strain that you found is a polyoma virus. So since the virus is a polyoma viridae, what's the type and form of the viral genome nucleic acid? Okay, so polyoma viridae, what is its genome like? Well, it's a double-stranded circular DNA, and it's monopartite. Okay, now why, if you watch my video or you watch the lectures from Dr. Samuels, why is it considered mini chromosome? Simple, because it wraps its double-stranded DNA around the same histones that cellular proteins wrap their DNA around. So essentially, this polyoma virus is a miniature chromosome. It's just like our own chromosomes because it wraps its DNA around. Because the virus wraps its DNA around the histones, you have a mini chromosome. Okay, so assume that you need highly purified. Basically, you need to isolate this virus. So this goes a bit into a lab technique. This was covered in lecture. If you look back through lecture notes or you rewatch lecture or whatever, there are steps to it. So you can try and look back towards it. I can't really draw it very fast. We'll use paint. Okay, so I hope this helps. Basically, you start with your lysate. Lysate is basically another word for a solution of virus. Okay, you put that through salt water 1 molar solution at 4 degrees Celsius, and you centrifuge it. Oh my goodness. I really wish I had a touch display right now. Okay, so you centrifuge it. And what you'll get is, the lysate is basically lysed cells plus virus. And what will happen is that the cell's DNA will be in the pellet, right? So that's like the stuff way at the bottom of your centrifuge tube, whereas your virus, I'm gonna use quotation marks, virus DNA is in the supernatant. Blah blah. Okay, I can't. And from there, what you're gonna do is do ultra centrifugation. Ultra centrifuge, blah blah blah. Okay, so that this is ultra centrifugation. From your ultra centrifugation, you need to do this ultra centrifugation in ethidium bromide. And what that will do is that it will separate based on the different forms of the viral DNA. So this will be form 2, and this is going to be form 1. So look back in one of my videos, or look back in your lecture notes, or look back in Dr. Samuel's lecture, form 1. And you'll see how I explain form 1 and form 2. Long story short, form 1 is in the super tight, super helical, super wrapped, super condensed virus genome form. Form 1 is when the viral genome is very condensed. Form 2 is when it's much more relaxed. And what happens is the ethidium bromide binds preferentially to form 2 and subsequently makes form 2 less dense than form 1. So, going back to question 4, you need a highly purified UCSB 2001 genome nucleic acid. You need to purify it. Okay. Free from contaminating cellular DNA. How do you do this? The super messy picture. Okay, so yeah, take the cells with the virus, lyse it, and then put that lysate through sodium chloride 1 molar, 4 degrees, centrifuge it. Then take the supernatant, centrifuge that in ethidium bromide, and now you have pure DNA. Okay. B. Alright, so blah blah USB, blah Burke Sharp, more stuff. You can probably read this faster than I can speak it. Important to note that we use S1 endonuclease and XO7 exonuclease. So first question is, what's the combined length of the exons of the spliced mRNA? Explain why. Okay, so this is background knowledge you have to have from discussion section. S1 is an endonuclease, and it specifically only cuts RNA exons. Okay, S1 is an endonuclease that cuts RNA exons. Well, I mean, more like just anything that has RNA. Obviously, yeah, anything that's an mRNA implies that it came from exon, duh, right? Well, no, not well. I take that back. S1 only cleaves. Let me rephrase all of that. S1 cleaves only single-stranded mRNA. XO7 is an exonuclease. It doesn't cleave. It doesn't really split a DNA or an RNA molecule into smaller bits. S1 only cleaves single-strand RNA. So if you have DNA, it doesn't matter. So if you use some reasoning, you can find out what the combined length of the exons of spliced mRNA is. So right here in the S1 gel, we see that there are two lines at 700 and 300 nucleotides. This is their size. So you add them up, 700 plus 300, that gives you 1,000. So that's the answer Y. Or that's the answer here. 1,000. The combined length of exons are 1,000 nucleotides. But it asks you to explain why. Now here's the reason why. It's because by looking at the data of S1, it is implied that the two fragments that you have there are fragments that show up from cut up mRNA. And this mRNA is indicative of the exons of the DNA, because the mRNA here, in this problem, it was implied that it was already spliced, that it had already gone, when you're going from DNA into RNA, you have to splice out the introns. And then you can make your mRNA fragments. So if S1 is the only thing that can cut these already spliced RNA fragments into smaller pieces, then this S1 shows you what the total length of the mRNAs mRNAs are, and that can give you insight into what the length of the exons are, right? Right, there's 700 nucleotides worth of mRNA here, 300 nucleotides worth of mRNA here. Another way of thinking that is there are 700 nucleotides of, in a way that is 700 nucleotides of exon and 300 nucleotides of exon. So that's why. It's because the S1, the data from X1 gel gives insight, it implies the length of your exons because it only cuts mRNA. What is the length of intron RNA? Well, you have your exo 7, which is an exonuclease, so it basically can't do much to your to your nucleotides, whether those nucleotides are DNA or RNA. So this line right here is your total genome size. So you know that 1,000 of the nucleotides are exons, and right here it says that the total size of your sequence is 1,800. So 1,800 minus 1,800 worth of intron and exon nucleotides minus 800 nucleotides of exons, or 1,000 nucleotides of exons, that means that gives you 800 nucleotides left of intron RNA, and you explain why. Okay, so next question. Highly conserved nucleotides are often found, blah, 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 at the splice junctions. This is something you have to memorize. The answer is, GU and GU and AG. That's the answer. Actually, let me just check. Yep, GU and AG. Done. Next question: the study of viruses that provide significant insights regarding parameters that can be important in determining efficiency of translation. Okay, so this question probably taken from the Rio Verde lecture. And this one is saying, testing your knowledge to see there are two answers. The first answer is in the context of the AUG start sites. And the next answer is the secondary structures on the mRNA. So if you have secondary structures on the mRNA, for example, a stem loop structure, then that will decrease translation. Now, the other answer you could have put is in the context of the AUG. So If your AUG is surrounded by purines, then the translation is going to be more efficient. If it's surrounded by pyrimidines, then the translation would be inefficient. Okay, so this one I'm just going to read off the blanks. So A is sialic acid and attachment. B is orthomixoviridae and rioviridae. C is hemagglutinin. D is pathogenicity. You can also put the term virulence. I prefer pathogenicity. E is polyadenylation. F is pandemic. G is papilloma and polyoma. You can also put orthomixoviridae because I believe 2 of the genes of orthomixoviridae, I think it's like the M2 and something else, maybe NP, are spliced. But I prefer papilloma and polyoma because they're DNA viruses and they undergo a lot of splicing. H is glycosylation and hemagglutinin. And I, this question, I thought it was Dr. Samuels asking what is Dr. Samuels' favorite virus, but it wasn't. This question is just, apparently this was given as a freebie at the time, and you just put down the virus that you like best. Personally, I have 3. Polio, because it was the first virus we learned, it's super tiny, and it has a lot of medical impact, health impact on the world. Next one is SV4, so polyoma viridae, because it revealed a lot of truths about cell genetics, eukaryotic genetics. But personally, I really like all 3 of them, but I think if I had to go with one, I put down influenza, and it's because that's still that virus has a lot of impact on people still to this very day. Okay, so question 6, almost done, guys, bear with me. Question 6A, influenza, blah, blah, 3 types. Okay, so influenza is, you guys know that it's classified into groups A, B, and C. Well, what this question is asking is, how do you get into those 3 groups in the first place? Well, in order to classify into groups A, B, and C, you first have to classify according to nucleocapsid protein and a matrix protein, so NP and M. And then, those classifications, the A, B, and C, have their own sub-classifications. And it is here that we then look at the hemagglutinin and the neuraminidase. So that's how you would answer that question. So you put down like NPM first, and then subtypes of A, or subtypes of those main 3 groups, influenza groups, that would be classified by HA or NA. Okay, so question B. Influenza viruses can be classified into subtypes based on their surface proteins, or its function, and neuraminidase protein during a single cycle. Okay, so just basically say, what does HA do? What does NA do? HA functions in binding and fusion, attachment of the virus to the cell, and fusion of the virus into the cell. And then NA, neuraminidase, is responsible for viral release, and it does this by upon leaving the cell, the virus has to cut the bonds between a sialic acid and a hemagglutinin. So that's how you would answer that question. Okay, vaccine flu zone, blah, blah. So this one is saying, how would you make recombinant hybrid Texas Nanchang virus strains? This question, honestly, if you really want to find out for yourself, look back at the flu lecture. That is the most clear, and there's a slide on it. I don't have it pulled up right now, but there's a slide on it. Pull it up. It'll help a lot. But essentially what you're going to do is that you can use either of the Texas strain or the Nanchang strain as a background strain. And that background strain, you take all the 8 different gene segments, and then you put in and you mix that with some of the capsid proteins. But you have to be careful in that the genome segments, everything except your hemagglutinin and your neuraminidase can come from background. So what I mean by that is, let's say you take the Texas strain as your background, then your PB1, your PB1, your PB2, your NS1, your NS2, your NP, and your PA can come from the Texas background strain. And your neuraminidase can come from a background strain, right? Because you want the neuraminidase as a Texas strain. But you have to make sure that your hemagglutinin gene comes from the Nanchang strain. And vice versa, you use the Nanchang strain as the background strain, then everything can come from Nanchang except for neuraminidase. You have to make sure that the neuraminidase comes from Texas. So once you have that set up, you then take those and you infect viral cell lines. Right, you infect viral cell lines, and then you take those viral cells that have been infected, you turn them into a lysate, and then you infect MDCK cells, and then you take the MDCKs that were infected by the viral lysate, which were infected by your mixture of genes before that. You take the MDCK infected cells, and then you get your recombinant cells out of them. And then you do plaque purification. Not too much detail there that I can give you, but essentially you do plaque purification, and the end result is genetically pure recombinant. Okay, so I hope that helps. I'll be getting this video out as soon as possible. Thanks for watching, and good luck on midterm tomorrow.